Alzheimer's disease originates in the hippocampus of the brain. In order to understand Alzheimer's disease, we need an understanding of how the signaling pathways within the brain work. For this, we look to WNT signaling pathway within the brain's neurons. In the normal chain of events, the WNT protein is produced in the endoplasmic reticulum of cells, including nerve cells, and transported across the cell membrane into the synapses between nerve cells. WNT is the ligand of the signaling pathway that transmits the signal from one cell to another. WNT travels to the adjacent neuron and is received by the frizzled receptor, which is a G protein coupled receptor, and the co-receptor LRP6, low-density lipoprotein receptor-related protein 6. The reception of the WNT ligand causes disruption in the complex of APC, axin, and GSK3 beta by binding axin to the LRP6 protein. This leads to the activation of the disheveled phosphoprotein, which inhibits the GSK3 beta enzyme. This series of events stops the degradation of beta-catenin. The beta-catenin can then travel into the nucleus and affects gene transcription by acting as a co-activator with the TCF, T-cell factor. This activates the transcription of target genes and inhibits the transcription of the BACE1 enzyme. BACE1 is partially responsible for the creation of plaques, which lead to Alzheimer's disease. In Alzheimer's disease, there is a noted lack of LRP6. Without enough LRP6 proteins, WNT signaling will be severely impaired. Without WNT signaling, the beta-catenin within the cell will be degraded and cannot travel into the neuron to help with the transcription of genes. This stops the BACE1 transcription from being inhibited. The BACE1 enzyme created as a result of that affects APP, the amyloid precursor protein, and causes the creation of amyloid beta. Amyloid beta will form oligomers that build up into plaques around neurons. Amyloid beta oligomers block ion channels and cause a buildup of calcium ions. Decreased WNT signaling also stops the inhibition of GSK3 beta, which then assists in the hyperphosphorylation of tau. Hyperphosphorylated tau contribute to the creation of neurofibrillary tangles. The plaques reduce activity between synapses, and neurofibrillary tangles become toxic, which can kill the cell. The resultant reduced signaling will cause the memory impairment we observe in Alzheimer's disease.